Hi, I'm Priya Duggal, and I am going to be the next speaker. I know that you were expecting Charles Chu, but there was a slight change in the schedule. So hopefully you can all hear me, and I will begin. So I'm going to talk to you today about host genetics and acute flaccid myelitis. And I want to thank the organizers, SRNA, and Dr. Pardo and his team for everything they've done to bring so much attention to AFM, but also this incredible symposium that we've all been a part of for the last several weeks. It's just been a wonderful learning experience. So why do we actually care about host genetics? So if we can understand why people have different outcomes, we'll be better able to understand the immune response and eventually exploit this understanding for therapeutics and potentially for a vaccine. So our goals here are still to really better understand disease so we can prevent and treat. And I start here because I think when people think about host genetics, they think that our goal here is genetic therapy of some sort. But really, it's to try to get an understanding of the immune system, much of what you've heard of today, but taking a slightly different perspective on the host. So the things that we do to look to see if genetics might be playing a role, just off the surface, looking at the epidemiology of disease, is to see if there could be some disease heterogeneity that's not explained by other risk factors, so not explained by things like sex or age or comorbidities. If there's familial aggregation, so if we see a specific disease aggregating within a family, and you can imagine that for something like cardiovascular diseases or cancer, where we see that things aggregate within a family. But this is a little harder to assess with infectious diseases because of transmission, because we also know that things transmit within a family. But we'll talk about some examples of the way that we can do that. And then we also want to consider, when we think about the role host genetics may be playing, is the role that pathogens may be playing, whether it be by dose or their environment. And if they're the major player or the major driver, then we suspect that host genetics will not be playing that major role. And then we want to see there's also similarities to other diseases in their pathology. So when we think about heterogeneity, the first question that really comes to mind is, does it really matter with an infectious disease because everyone gets it? And I just want to bring your attention to some of the heavy hitters like tuberculosis and hepatitis B, which are known to infect about a third of the world's population with either the mycobacteria or the virus. But only about 10% are actually going to develop clinical disease for mycobacteria, and only about 10% are going to develop chronic disease for hepatitis B viral infections. So despite the fact that people are getting infected, not all of them are having disease. And then similarly with HIV/AIDS, the average time from HIV to AIDS is about 10 years, but the range is 2 to 26 years. So what is that variability, that heterogeneity that's introduced with disease progression? And this is, of course, without treatment, but even with treatment in the post-heart era, we see that heterogeneity exist. So when we think about familial aggregation, as I mentioned before, I found this really nice paper that was published in 1938 by Wade Hampton Frost, who's really considered the grandfather of epidemiology in many ways, writing on the familial aggregation of infectious diseases. And in this, he's really talking more about transmission within a family, which is where we often think about with infectious diseases. So secondary attack rates, for example. And what I'm really asking is this question of, within a family or within a pedigree, are individuals who are more related to each other more likely to also have outcomes? And one way we can do that is looking at monozygotic and dizygotic twins. So with monozygotic concordance rates, those are individuals we consider identical twins. They share 100% of their DNA, compared to dizygotic, who are siblings in equivalent, but had shared the same womb. We would expect that if something was more genetic, it would have more concordance in the phenotype with the monozygotic and the dizygotic. And this is just some examples of infectious diseases where that's been true: leprosy, hepatitis B viral infection, and TB. And then we can quantify that and get an estimate of how much we think genetics is actually contributing to disease. We call that heritability. And for something like HBV, we think that's roughly 60%. And for something like TB, it would be 70%. So in these examples, and we could do the same for other diseases, we're getting at this idea of what is this contribution, and is there something worthy of us looking at for genetics? And then, of course, we can't completely control dose. We can do challenge studies, which are well-controlled examples of giving someone a specific pathogen. But otherwise, it's not really ethical for us to be controlling the dose or giving people certain infections. We have a history of having done that and knowing how wrong that is. But we can also, at the same time, learn from our mistakes that are natural experiments. And this is one that happened in 1926 in Lübeck, Germany, where 249 babies were injected with the same live dose of tuberculosis instead of the BCG vaccine. The babies were too young to have significant prior exposure to a mycobacteria, and they had not previously been vaccinated to BCG. So in this case, they're all getting the same strain and the same dose. And what we see is that 76 babies died and 173 babies survived. So this heterogeneity is existing again, even when we control for the dose or that specific pathogen. And so then that leads us to this idea of the next thing on my list of thinking about where other pathologies might fit, and where it's natural for us to go is the poliovirus, and poliomyelitis. And so these are headlines that some may look reminiscent of today, actually, when we think about quarantine, but really reflect what happened with polio, as there was large pandemics and epidemics that occurred here in the United States. And of course, as polio eradication is just within reach now, after so many years of us having to fight it, in 2014, a new disease emerged, and that's the focus, of course, of this symposium, this mystery paralysis, as they call it, acute flaccid myelitis. And so I put these slides together really to get a sense of what could we learn from polio that would help us with AFM, as we're still sort of in the early days of case numbers for AFM. So what we know about poliovirus is it comes from the Greek words polio and myelon, representing gray and marrow. It affects the gray matter, as does AFM, and there's extensive damage to the anterior horn cells of the spinal cord, which causes limb paralysis. Its incubation period is similar to enterovirus, and its widespread muscular atrophy is similar to AFM. But what we see is that poliovirus is part of the enterovirus family, which is part of the picornaviridae family, and you heard about that earlier in some of the talks today. In 1894, the first U.S. localized paralytic polio epidemics began to appear in the United States, and by June of 1916, New York had a huge epidemic of more than 27,000 polio cases and greater than 6,000 fatalities. And in the 1940s and 50s, polio cases soared, 15 to 20,000 cases per year. And then, of course, as we're all familiar with, the vaccine changed everything. And as we just heard in the discussion session a few minutes ago from Ken Tyler, it wasn't therapeutics. It was the vaccine that made the change. And it's really what we're all aiming for. And in 1 year, the number of cases went from 58,000 to 5,600. But in that process of us being able to stop polio in its tracks, we never really learned what caused polio paralysis. So we never really understood what made those children different, what made them that 1 in 100,000 or 1 in 200,000 child that occurred. And if we had figured that out, I think we'd be in a very different place with AFM than we are right now. So again, if I think about the factors that we think about when we think about genetic epidemiology, what can tragedy teach us about dose and virus? And so we heard some talks about the virus itself and the evolution of that virus, which are very important. But in any given year, in a family, what could we also learn about it? And this is an example, another tragedy where the polio vaccine was administered that was not inactivated to school-age children. The same active virus was administered, but we clearly see different outcomes. So 200,000 children were injected, 40,000 ended up with infectious symptoms, and 200 had paralytic polio, and 10 children died. So again, if we're getting the same similar infectious dose of the same live virus, why is it that everyone didn't get infected? And why did not everyone get paralysis? And these are the same questions I think we should be asking about AFM. So what can we learn about familial aggregation from poliomyelitis? Well, there have been some great twin studies, and I highlight one of them where they evaluated what I presume were monozygotic twins. At the time, they didn't have testing to prove that they were the same. And 5 out of 14 monozygotic twin pairs had paralytic polio for a rate of 36%, compared to the dizygotic twins where the concordance was only 6%. No parents had a history of poliomyelitis, consistent with what we see with AFM, and there was no known intermarriage. So these authors suggested that it was likely a recessive gene with relatively high frequency in the population that was driving polio. And I think it gives us a lot of information about what we need to be thinking about with AFM, where we have very few numbers overall and an inability to really look at monozygotic versus dizygotic twins at this time frame. But we also know that even from polio, it wasn't 100% genetic. So it's not what we're driving at. We don't believe this is a primary immunodeficiency or a primary neurologic outcome, but we know that genetics is probably playing some role in it. We also learned from two different studies by Darren Snyder and by Wyatt about how it did aggregate in families over generations. So in one, polio cases in West Virginia, 29 cases over 50 years could be attributable to 25 related families. So on the surface, it looked like 25 different families, but when investigators went in deeper, they realized that all 25 families were actually related into one very large pedigree, and all the cases in that area were attributable to that one family. And then similarly, in a study of individuals from Malta, there were 1,000 cases of poliomyelitis over 60 years that were traced. And you can see that most of the cases actually occurred, 54%, between siblings, first or second degree cousins. But what was most fascinating was that there were 13 pairs of siblings where they both had paralytic polio, but this younger sibling wasn't even born at the time when the first sibling had the paralysis. So this wasn't about transmission within the family. This was likely about a genetic predisposition to having that particular paralysis. And so then when we think about AFM, with all of those factors, is there actual disease heterogeneity in AFM? And we've already heard from several speakers that there is. And some of the usual suspects of the contributors to that disease heterogeneity outside of the virus and the host genetics is age, sex, and co-infections. So with age, we're dealing with children. We don't see waning immunity that would have playing a role to make you more susceptible. In terms of sex, we see that more children are affected and they're likely prepubescent when they are affected. So it's not likely a huge skewing towards individuals based on hormonal differences, which we often see when we look at infectious diseases. And then, of course, a big player would be co-infections and comorbidities. And what's fascinating with AFM is that we actually don't seem to have children with underlying health conditions, whether they be immune-related or neurologic, that are the ones that end up paralyzed. But instead, parents report that their children were very healthy overall, and yet they had it. So some of these usual suspects are also not playing a role. And so as a result of that, we designed a genetic study where we actually do a case family design. We highlighted the fact that we know that the family members are really important to us here because we want to know that exposure was similar within the household. We also want to be able to evaluate siblings because to date, no two siblings in a household have yet been identified. And so we have households where a case has been affected, a child, and their siblings are all unaffected, despite the fact that most parents will report that all of the family members had the same viral illness that preceded the paralysis. And so the key here is that in using this family study, we're able to know what's going on in the family, and the controls, the siblings and any other household members, become those individuals who had been exposed presumably to enterovirus and yet didn't develop that paralysis. And that exposure point is really key for us because otherwise we're just saying, did you develop paralysis or not, without considering the virus. And then we whole exome sequence, and in some cases we could whole genome sequence all the first degree relatives living in the household. So that's the parents and any siblings. We have more than 100 AFM case families enrolled in our study through December of 2019, and the mean age of our child cases is 5 years. We also have 4 adult cases that have been enrolled with a mean age of 30 years. We do have more boys than girls in our study, but that's not necessarily reflective of true disease. That's just who's in our selected study. You can see from the map on the right, we represent a good portion of the United States. And we have a variety of limb paralysis involvement, from full limb paralysis to 1 limb or up to 3 limbs. And the self-reported race ethnicity of individuals does vary. It does seem at first that we have a majority of individuals who are self-reported white, but I will say that actually 42% of our population consider themselves other or mixed. And so what have we actually found? So I'm happy to say that we've done some initial analysis, and it's been a long haul, in part because we have to be able to identify and get these cases enrolled in our study, and then we have to do the sequencing, and we're not always flush with money to make that happen. But we do note that AFM cases have more family members with polio than is expected, which is exceptionally intriguing for us. Because they seem to be enriched. We've also evaluated what we call de novo mutations. Those are new mutations that occur where neither the parent has the mutation, so they didn't pass it on, something brand new occurred in parent-child trio. So we need the parents to evaluate this. And we see an enrichment of loss of function de novo mutations in some of these pedigrees. So we identified 14 of 17 cases with a de novo mutation, and 11 of those 14 had a median of 4 de novo mutations, and they could range anywhere from 1 to 14 that actually occurred. What was most interesting is that in 5 of the 11 cases, we identified 18 de novo mutations. They were all frame shift mutations, and they all caused a loss of function, and they spanned 17 different genes. And there seemed to be a large difference between what we would have expected, which is 1 loss of function de novo mutation, versus the 18 that we actually observed. Unfortunately, we didn't see a smoking gun where there was a clear common gene or a clear known pathway that emerged among these first 17 cases. But replication studies are underway with an additional 25 cases and their families. And we also plan to evaluate our polio survivors so that we can see what's happening with those individuals. So what can we do, and can genetics help? I think we've heard a lot about the concern about acute flaccid myelitis emerging into something much larger, either this year or next, and I think that's what's foremost in our concern. Is there a way that genetics can help to identify who might be at risk, either within pedigrees that have already been affected or with new families? Can we also figure out who would be most at risk for certain types of paralysis? And then, of course, we know that poliomyelitis was stopped with a vaccine. So AFM may also need to be stopped with a vaccine. And can genetics help to enhance a potential vaccine by the knowledge that we gain from it? And so these are just some folks I'd really like to help acknowledge, specifically our original team of Aaron Millstone and David Thomas, where we started the study in 2014, the expertise of Carlos Pardo, Matthew Elric, Olwen Murphy, and Tom Crawford on all of the neurology. All of our cases are reviewed blindly by multiple neurologists, and the research coordinatorship of Hannah Manley and Ani Vaskurchian, who've helped us get so much of the information that we need from our participants and our families. And then on the genetic side, Christian Valencia, a doctoral student with me, has done a tremendous amount of work in trying to work through these families and try to understand the de novo mutations. And then, of course, all of the physicians and families that we work with overall. And I'm happy to introduce next Christina Cassetti, who's going to talk to you about AFM, enteroviruses, and vaccines.